The microscope is used for observation of cell bottle, flask and multi-layer culture dishes. The inifite optical system offers high-clarity observation. It is also can be applied to the cell factory immediately. See the cells in the bottom 2 layers culture dishes. Components introduction. Microscope body, phase contrast plate, head, eyepieces, sample holder, objective, light source and arm. The compact structure is easy to use and stable working. The metal material is anti-shocking. 9-watt LED lamp lighting, instant on and off to save time and energy. It is long working life, less maintenance needed that save cost. The 45 degrees inclination head is comfortable for human using. Adjustable pupil distance and diopter. Wide field of view eyepiece in high eye point is friendly to eyeglasses wearing users. Here are long working distance objectives for bright field and phase contrast observation method. Compatible to study the big and tall object. That is the stage and hand wheel controls is moving in X and Y direction. There are focusing knob on both sides. Outer ring for coarse focus, inner ring for fine focus. Power button is here. The phase contrast plate supports 10 times, 20 times, and 40 times phase contrast. Enhance the observation performance of transparent objects, like live cell. Installation steps. Install bright field light and arm. Firstly, connect the light terminal, then screw in the 4 screws. Install objectives. Screw in the objective to the nosepiece one by one. Put on sample holder. Stuck in the sample holder to the stage. Connect microscope head and eyepiece. Push in the head into the observation tube. Fix it by locking the screw on the right side. Insert in the eyepiece to the tube one by one. Install phase contrast plate. Straightly push in the plate to the slot softly to feel each position.